So in terms of visualizing aspects of neuroanatomy, both gross and fine, you actually need to have ways of making these certain structures or even connections between structures visible to the eye. I mean, we only have, a certain range of perceptual apparatus ourselves in order to be able to, detect or appreciate, aspects of neuroanatomy. And so there were initially a number of stains that were developed to help to, latch onto certain structures and make them visible, under the microscope to the naked eye. And your book talks about a stain called the Nissl stain that Franz Nissl developed, which binds to ribosomal RNA. So it really kind of identifies like the endoplasmic reticulum of cells, so where the the process of translation, taking that messenger RNA and feeding it into the ribosome and generating the protein machines like the channels and the the pumps and the transporters and the receptors, etc., is occurring. And one stain that's particularly critical for sort of recognizing or identifying neurons is something called the Golgi stain. And Camillo Golgi actually developed this stain. It's a silver stain. He was a neuroanatomist and also a rather excellent artist. And he would actually observed his stained neurons. And this was a silver stain that got picked up selectively by some neurons and not by others. So it took advantage of some kind of transport mechanism that was present in some neurons but not others. But if it got in, it would be distributed to all the different processes in that neuron. So it would actually stain, all the dendrites and the cell body and the axon collaterals and the terminals. And he was an artist, this Golgi, and he would observe, the stained sections of neurons that he saw under the microscope, and he would draw them. And this is actually, from your overhead, these are some of the drawings. I'm sorry, the light drops dramatically whenever I do this. But this is basically shows you, like, it looks like a massive sort of interconnected wires with little swellings in some locations, that represent the cell bodies. And Golgi himself actually thought that this actually did literally represent a massive interconnected wires, that it wasn't that the cell doctrine, which states that tissues are composed of individual sort of units that are known as cells, right, did not apply to the brain, because he didn't see what looked like a normal sort of spherical or oval cell, right? He saw these sort of processes that sort of intersected all over, and he couldn't really detect the location of the gaps or synapses between the axon terminal of one neuron and, let's say, the dendrite of the next. So he actually thought that the brain was what he termed a syncytium, which means a massive interconnected wires, actually. And he published these drawings. And another well-known neuroscientist, Santiago Ramon y Cajal, in Spain, actually thought he saw independent cells, just unusually shaped cells, with extensive, dendritic arbors, and long processes or axons that extended away from the cell body. And ultimately, the two of them won the Nobel Prize. Golgi came around to Cajal's way of thinking and agreed that the cell doctrine also applied to the brain. But this stain was very important for, in terms of visualizing, the various structural aspects of individual neurons. Now, another class of chemicals that you need to know are what are called tracers, because one of the important pieces of information you can get from the study of neuroanatomy is what is connected literally to what. You want to be able to trace some of these networks that we're going to be focusing in on when we start talking about cognitive behavior and the underlying kind of neural architecture. So tracers are chemicals that are injected into an area of the brain, and they can then get distributed elsewhere. And so you can have, and so they can give you a sense of what's linked up with what. And in terms of tracers, there are two kinds that I'd like you to sort of focus on. One is called a retrograde tracer. So a retrograde tracer is a chemical that'll be injected into an area of the brain, right? And it'll be picked up by the axons, the axon terminals of the neurons that are in that location, and then it'll be transported back, in a retro direction, and then stain the cell bodies that gave rise to, that projected those axons forward into that region. So what it will do, a retrograde tracer will stain the area that is projecting to the area that you injected the tracer into. So it gives you a sense of where inputs into a particular brain region are coming from, because it goes back and stains the source, right? And the other kind of tracer that you should be aware of is called the anterograde tracer. And an anterograde tracer is injected, let's say, to the same area, but it'll be picked up by the dendrites and the cell bodies of neurons in that region, and then be transported out to the axon terminals. So it'll help to stain the projections from a particular area. It goes, antero means in a forward direction, right? So let's give you an example. Let's say you were going to be injecting a retrograde tracer into the lateral geniculate nucleus, which is, of course, the thalamic nucleus that's processing visual information. Well, the projections to the LGN are coming from the retina. So if you put a retrograde tracer into the LGN, it's gonna be picked up by ganglion cell axons, and go back and stain the ganglion cells that are located in the retina. If you inject an anterograde tracer into the lateral geniculate nucleus, well, it's going to be picked up by LGN, dendrites and cell bodies, and it's going to, be transported out to the axon terminals that are located in primary visual cortex in the occipital lobe, right? Now, the last type of tracer I want you to be aware of is something called a metabolic tracer. And a metabolic tracer is some kind of visible tracer. So typically it's a radio-labeled substance. Something radioactive is linked onto it. It could also be something that fluoresces, actually, under certain kinds of light. But it's attached to some chemical that's going to be involved in the metabolic activity of cells. So it's sort of a functional tracer. It'll help you identify areas of the brain that are more active under certain conditions, because it's going to be utilizing this chemical that's involved in metabolism. If there's more activity, you're going to get more of this radio-labeled chemical collecting in that area. And so I've got an example. This is a study from the days when there was less sort of institutional review board kind of overview of experimentation. These were early days also in terms of trying to understand the structure and function of, let's say, the visual system. So this was an experiment that was done in monkeys, and macaque monkeys. But basically, they presented these monkeys with this visual image on a screen. So this is the left visual field presentation. This is the right visual field presentation. And you can see these were all sort of shimmering lights that were going around in this particular pattern. And they injected a particular kind of metabolic tracer. I think it was radio-labeled glucose, actually, which will be picked up and utilized more by cells that are carrying out more metabolic activity. And they sacrificed the macaque after showing it this particular image. And when they splay out the visual cortex, you can see the actual pattern of what was being presented in the world out here, right? The macaque's retina is sort of displayed on primary visual cortex. But what's interesting is that the area that corresponds to the fovea, the monkey was focusing right here, gets a lot more representation proportionally than the area that's out in the periphery. You can see this is sort of expanded out at the level of the fovea. So this gives you some idea of the sort of brain architecture that underlies the homuncular organization of any kind of sensory cortex, right? Where you get more representation for certain stimuli versus others. And in the case of the visual system, obviously, you have a lot more of those tightly packed cone cells that are located in the foveal region of the retina. And it turns out that you get a lot more brain space that is devoted, cortical space in primary visual cortex that's also devoted to that region of the visual world.